Hello viewers, I am Dr. Nazia Sharmin. Hope you all are doing well. Today I will discuss about the process of carbohydrate digestion. The major foods on which our body lives can be classified as carbohydrates, fats and proteins. They generally cannot be absorbed in their natural forms through the gastrointestinal mucosa and for this reason are useless as nutrients without preliminary digestion. That is why in my today's video, I will discuss the processes by which carbohydrates are digested into their small enough compounds for absorption. Here are the key points. First of all, definition, then basic mechanism of digestion, major dietary carbohydrates, key enzymes needed for carbohydrate digestion, and brief process of digestion, and after all this, final end products of digestion. Now I am describing the key points one after another. First of all, definition. Digestion is a physiological process by which large complex food particles are broken down into their simpler forms suitable for absorption. Or we can simplify, digestion is a process by which the ingested food particles are broken down to smaller molecules suitable for absorption or assimilation aided by digestive enzymes. Now, basic process of digestion, that is hydrolysis. Hydrolysis means breakdown of larger particle to smaller one with addition of elements of water. We have two elements of water, one is hydrogen ion and another one is hydroxyl ion. For example, maltose is an disaccharide. With addition of elements of water, this maltose become broken down into two molecules of glucose. The third one is major dietary carbohydrates. Number 1, that is starch, then lactose, sucrose, glycogen and cellulose. Starch mainly consists of amylose and amylopectin, found in almost all non-animal foods, particularly in potatoes and different types of grains. Lactose mainly found in milk, that is why lactose is known as milk sugar and sucrose commonly known as table sugar. Some general information about amylose and amylopectin. Amylose and amylopectin both are homopolymers of glucose, but amylose is unbranched and linked together by alpha-1,4 glycosidic linkage, but amylopectin is branched in structure. In straight chain, they are linked together by alpha-1,4 glycosidic linkage, and in branched chain, they are linked together by alpha-1,6 glycosidic linkage. We have some other less important carbohydrates like alcohol, lactic acid, pyruvic acids, pectins, dextrins, glucose, fructose and pentose sugar. Now the fourth one is principal enzymes needed for the digestion of carbohydrate. In saliva, we found salivary alpha-amylase or ptyalin. In pancreatic juice, we found pancreatic alpha-amylase. In succus entericus, there are maltase and isomaltase, lactase, sucrase and alpha-limit dextrinase. Here the list, but not the last, that is process of digestion. Carbohydrate digestion starts from mouth. When the food is chewed, it is mixed with saliva, which contains the digestive enzyme ptyalin, secreted mainly by the parotid glands. Then salivary alpha-amylase hydrolyzes 20 to 40% of boiled starch into the disaccharide, maltose and other small polymers of glucose that contains 3 to 9 glucose polymers, namely maltodextrose and alpha-dextrin. From mouth, food enters into stomach. No carbohydrate digestion occurs in stomach because of high acidity of gastric juice inhibits the action of salivary alpha-amylase. Starch digestion sometimes continues in the fundus and body of stomach for as long as 1 hour before the food becomes mixed with stomach secretions. Then activity of the salivary alpha-amylase is blocked by acid of the gastric juice because the amylase is essentially non-active as an enzyme once the pH of the medium falls below about 4. Within 15 to 30 minutes, acidic stomach contents passes into the duodenum and mixes with pancreatic secretion which contain pancreatic alpha-amylase. This pancreatic alpha-amylase hydrolyzes the remaining 50 to 80% of boiled starch and all the unboiled starch into maltose, maltotriose and alpha-limit dextrins. Virtually all the carbohydrates will have become digested. Here is the starch. This is the straight chain structure of starch and here is the branch chain structure of starch. By the enzyme salivary alpha-amylase and pancreatic alpha-amylase, starch broken down into maltose is a disaccharide consists of 2 glucose molecule. A oligosaccharide consists of 3 glucose molecule, that is maltotriose, and alpha dextrins consist of several glucose molecules. After all this, carbohydrate digestion is completed in small intestine, producing monosaccharides by the 4 branch border enzymes, namely maltase, sucrase, lactase and alpha dextrinase, which digest all the disaccharides and small glucose polymers into absorbable monosaccharides in the following way. Maltose and maltotriose both are digested by the enzyme maltase. Maltose converted to 2 glucose molecule and maltotriose converted to 3 molecule of glucose. Isomaltose broken down by the enzyme isomaltase and produce 2 glucose molecule, alpha dextrin by alpha dextrinase and produce several molecules of glucose. Lactose broken down by the enzyme lactase produce glucose and galactose, and sucrose broken down by the enzyme sucrase and finally produce glucose and fructose. Here is the general overview of carbohydrate digestion. Starch by the enzyme ptyalin and pancreatic amylase broken down into maltose and 3 to 9 glucose polymer. Then these maltose and 3 to 9 glucose polymers are broken down by the enzyme maltase and alpha dextrinase, ultimately glucose. Lactose broken down by the enzyme lactase and produce galactose and glucose, and sucrose broken down by the enzyme sucrase and produce fructose and glucose. After overall digestion process of carbohydrate, here are the final end products of carbohydrate digestion, that are glucose, galactose and fructose. That's all, viewer. Now, thank you.